In this next video clip, we're going to start talking about some of the chemicals that we use for microbial control. So many of these things will be familiar to you. Now in general, so we talked about early in Chapter 9 in the first video lecture about the modes of action for physical and chemical agents for controlling microbes. So in general, they're going to affect the cell membrane, cell wall, proteins, DNA, some sort of metabolic processes inside cells, and some of these will impact more than one of those. And we've talked about how the various things that can affect those agents: temperature, pH, the exposure time, very, very important, how well these substances can penetrate through other materials that are present, other organic matter like blood or feces or saliva that might be there. Now, when you start thinking about chemicals, you've got to consider freshness and concentration. Some of these chemicals break down over time, so you've always got to be concerned about that. And concentration is important as well. It's recommended that you dilute a concentrated disinfectant 1 to 100 and use it at that concentration. It's very important that you follow those directions because, believe it or not, there are some things that don't work as well when they're more concentrated than the recommended amount. So you just have to be really, really careful with those types of things. Your chemical disinfectants and antiseptics tend to fall into several different categories, and those include phenols, alcohols, halogens, oxidizing agents, surfactants. Detergents that we use are examples of surfactants. Heavy metals, so things like mercury, for example, aldehydes like formaldehyde, and then there are certain gases that are used as well for chemical control. Some terminology. Germicides. Remember, again, a germicide is a substance that kills microbes, in general. We're not necessarily focusing in on one category of microbes. So it's really another term for microbicide as well. High level, intermediate level, low level. So pretty much means what the names suggest. If you have a high level germicide, that's something that you may be able to achieve sterility with if you have a long enough contact time. So it's going to kill all of your pathogens with a long enough contact time. It can kill endospores, so they can be sporicidal. So what would you use these for? There are certain instruments, things like catheters and implants, heart-lung machine parts. Those are things that are reusable, but you can't stick them into an autoclave, or you can't put them in a hot air oven because they're too delicate. But sometimes those things can be exposed to chemicals for long periods of time to achieve sterility. Intermediate level germicides. Now these are a lot of the disinfectants that we use on a more typical basis fall into this intermediate level germicide category. Now these, you can't feel confident that they're killing the endospores of bacteria. These are sometimes used to disinfect instruments that contact mucous membranes in a non-invasive manner, so they don't poke through the mucous membranes. Man, these mucous membranes, you have microorganisms there on a regular basis anyway, so the concern is you don't want to puncture through that surface. Sometimes, now you guys know a lot of if they're going to stick something down your throat and contact a mucous membrane there, a lot of those supplies are sterile anyway. A lot of times sterile, disposable types of things are used medically as much as possible. All right, then you have low-level germicides. So these are going to be your weaker ones, and they'll generally kill vegetative bacteria, not endospores, fungi, protozoans, some viruses, as long as you're using the appropriate contact times. These are going to be used if you want to clean furniture and electrodes, things like that, that are going to only contact the skin of a patient. They're not going to penetrate the skin. So those are kind of three general categories. What are some of our desirable chemical germicide qualities? So if you could come up with an ideal germicide, what would you want this to do? What features would you want it to have? Rapid action at low concentrations, that would be great. Low concentration. A lot of these are chemicals, if we as people get exposed to them in high doses for long periods of time, they're not too good for us either. So ideally, you would be able to use a low concentration for a short period of time. You want it to be soluble in water. That means it dissolves in water. You can form an aqueous solution. Now, some things you can't. Some things we use as disinfectants and antiseptics have to be dissolved in alcohol, and that's called a tincture. Tincture is a term for a solution of some sort of chemical that has been dissolved in alcohol rather than water. So think about it like when you take cough medicine, for example. You know, it tastes terrible, it burns your throat. That's because that chemical drug that's in the cough medicine doesn't dissolve well just in water. You got to have some alcohol there to help that dissolve. So something like a cough medicine is often going to be a tincture rather than an aqueous water-based solution. It'd be great if your germicide had long-term stability, so you can go to Walmart and buy some in 2010 and it's still working in 2018. That's not always the case, though, so you got to read labels, read expiration dates. That's very, very critical. Broad spectrum action. You want it to kill as many different kinds of microbes as well. Narrow spectrum would mean it would act on only a limited range of microbes. Broad spectrum, lots of microbes. Ideally non-toxic to human or animal tissues. That's just kind of common sense. That's not always the case, though. Surface penetration, you want it to penetrate surfaces if possible. Non-corrosive, non-staining. So if you're using a disinfectant on your kitchen counter at home or in your bathroom, you don't want it, if you're spraying a disinfectant in your shower, you don't want it leaving stains in the shower. You don't want it breaking down your kitchen counter. Those are ideal types of things. Deodorizing, it's good if we get rid of foul odors. And of course, affordable and readily available. So some of those things are just kind of common sense. All right, and again, this is table 9.8 from your textbook, which is emphasizing for you that concentrations and exposure times matter. So they're showing you some different chemical agents like chlorine, ethyl alcohol, that's ethanol, hydrogen peroxide, quaternary ammonium compounds. We'll talk a little bit more about those shortly. Ethylene oxide is a type of gas that's used as a chemical control agent. And concentrations over here, there are different ways of reporting concentrations, like ppm, parts per million. So that's used for substances that are very, very, very heavily diluted when they're used as a disinfectant or an antiseptic. And then over here you have your exposure times. A lot of things the concentrations are given as percentages. It's like ethyl alcohol, commonly used as a disinfectant at a 70% concentration. That means you would mix 7 parts ethyl alcohol to 3 parts water so that you have a 70-30, a 70%, 30% ratio. Look at hydrogen peroxide, 3% is your typical working concentration. So if you were making 100 milliliters of that, you would have 3 milliliters of hydrogen peroxide and 97 of water, so that hydrogen peroxide is in there at a concentration of 3%. Or ethylene oxide gas, its concentration is given in milligrams per liter. So those are different ways that concentration can be reported. All right, so let's talk about some of these different chemical control agents and the categories that they fall into. And there are going to be many different examples in each of these categories. And one thing I want you guys to do is take a look in your textbook, because your textbook has a very nice table that gives you an overview of different types of chemical control agents, and they have the table colorized to highlight things that are capable of sterilizing if you use them at appropriate concentrations and for proper exposure times, versus ones that you really can't use to achieve sterility. So take a look in your textbook. Take a good look at that table very thoroughly while you're doing your reading. So let's talk about some of these groups. So we've got halogens, and you learn about halogens when you take a chemistry class. That's a group of chemical elements that have related chemical properties in terms of how they react with other chemicals, and these include iodine, chlorine, bromine, fluorine. Those are all in the halogen category. So you're probably recognizing some of those as things you might use for controlling microbes. In general, these are going to be intermediate level, but with very long contact periods, you can achieve sterility with some of these in some instances. So because they're intermediate level, they're going to kill the things that we associate with intermediate level antimicrobial agents. So these can kill bacterial endospores, but again, that exposure time is going to be really, really chemical. Now, how do these work? Mode of action. And again, look at that table in your textbook, because it's going to give you some more details on modes of action of these different categories of antimicrobial chemicals. But they generally work by reacting with proteins. They either cause proteins and enzymes, protein enzymes to denature, or in some cases, these are going to be things that attach to the enzymes and block those active sites and prevent them from working. So iodine is often used as an antiseptic. So remember, antiseptics are used on living tissues, human or animal tissues like the skin. And a real common iodine-based antiseptic is betadine. We've all seen that for the brown stuff they rub on your skin. And again, just keep in mind, when they do that, they're not achieving sterility on your skin. Your skin is too sensitive to be exposed to a chemical for a long enough period of time to achieve sterility. The goal there is to reduce the number of microorganisms that are present down to a hopefully safe level. You're trying to lower the probability that a patient is going to develop an infection when you stick a needle through their skin. All right, some chlorine-based compounds. Now, you guys know that chlorine gets added to swimming pools to help lower the number of organisms that are capable of growing in the swimming pool. It's also used to treat your drinking water. Wastewater, like at sewage treatment plants, is exposed to chlorine. That's Cl2, two chlorine atoms bonded together. And what these compounds do, they combine with water and form hypochlorous acid. And it's the hypochlorous acid that's very chemically reactive as well, and can help with the mode of action for these particular kinds of compounds. In bleach, good old-fashioned bleach, sodium hypochlorite is the active ingredient in there. It's in there at a pretty low percentage because it is very chemically reactive and damaging to microorganisms that it comes into contact with. Bromine, I'm not sure that's mentioned in your textbook. That is kind of like chlorine, but bromine does not evaporate out of things like hot tubs as quickly as chlorine does. So sometimes hot tubs will have bromine put in there to control microorganisms more so than chlorine. And obviously hot tubs, that's kind of like an incubator. People are sitting in there with their body parts, so you're going to have a lot of microorganisms in a hot tub, so you want something in there that's keeping those microorganisms under control. And now you guys will never go into a hot tub again. Now, hot tubs are generally going to be safe. All right, another class of chemical control agents are considered to be oxidizing agents. So they do oxidation chemical reactions on important molecules inside Chapter 7. And these include things like peroxides, like hydrogen peroxide, ozone, peracetic acid is another one of these. And they're highly chemically reactive. You guys can remember talking about hydrogen peroxide and how damaging and chemically reactive it is with important biological molecules in cells back when we were going over that in Chapter 6. So these are capable of screwing up enzymes and other important molecules in cells. All right, so in some cases, some of these can be high level, so they can achieve sterility with high concentrations, long exposure times. You guys also may remember from Chapter 6, we talked about how hydrogen peroxide, for example, and other nasty oxygen-containing compounds are generated all the time through just regular day-to-day processes that occur in cells that get exposed to oxygen. So living things that are exposed to oxygen have to have enzymes that detoxify those types of things, otherwise they would not be able to survive. So oxidizing agents are very effective against anaerobes, because remember, anaerobes can't be around oxygen gas because they don't have the enzymes that detoxify the nasty oxygen-containing compounds. So some of these things are going to be very effective against anaerobic organisms that may need to be controlled. Hydrogen peroxide, all right, so your mama might have put hydrogen peroxide on a cut when you were scraped when you were a kid, and it bubbled, and you know, my mom always talked about how she just liked hydrogen peroxide. You know, it bubbles, it just looks like it's doing something, you know, but actually all that bubbling, if you guys remember learning about catalase is an enzyme that breaks hydrogen peroxide down into water and oxygen gas, H2O plus O2. So all that bubbling you're seeing is actually the catalase inside your cells breaking down the hydrogen peroxide. So it's probably not going to have any kind of long-term effects as an antiseptic. That's not saying it's completely worthless, but because you are applying a lot of it, so a lot of the microorganisms that are in a wound are going to be killed by the hydrogen peroxide, but it's not going to do anything long-term for you. But it is a very good disinfectant for surfaces. Actually, when students test this in our microbiology labs on campus, when they use hydrogen peroxide as a chemical disinfectant on some surface, they usually get really good results with it. You know, on a surface, you're flooding these microorganisms with it, and even if they're capable of detoxifying hydrogen peroxide, they're not going to be able to do that fast enough to make a difference. Aldehydes. So this is a group that includes things like glutaraldehyde, formaldehyde. You guys know formaldehyde is used as a preservative, you know, for things like lab specimens. Formaldehyde is used in embalming after you die. So these are very chemically reactive compounds, and the way they work is they cross-link biological molecules that are adjacent to each other together. So if you got protein number 1 here and protein number 2 over here, if they're close enough together, these aldehydes will cause them to bond or cross-link to each other. So basically you freeze the biological molecules in cells in place. That's why these are such good preservatives. You can have something like a cow heart sitting around in a jar in a laboratory in formaldehyde for a long period of time, and it's not really going to break down. It helps preserve the structure. So it's basically freezing these things in place, preventing these proteins from working anymore, and activates nucleic acids. Glutaraldehyde is used in hospitals, things like respiratory equipment, dialysis equipment, you know, stuff you can't put it has to be reused because it's so expensive, but you can't put it into an autoclave. Some of these things can be sterilized with long 10-hour exposures to glutaraldehyde. Formaldehyde is used in embalming, typically used at a 37% concentration, so 37% formaldehyde, 63% water. That's called formalin. Very irritating, though. These are not chemicals that you want to expose yourself to for long periods of time. How about some gaseous agents? So there are some things like pillows, mattresses, electronic equipment, some plastics, prepackaged supplies, things that you can't expose to heat, you can't get it wet, you don't have any other method for disinfecting or sterilizing. Sometimes you can expose these things to gases. So now these gases are very toxic, so they have to be used in a gas chamber, a closed chamber. And, you know, many of these are kind of explosive, so there are safety concerns with these as well. You have to be really careful. Ethylene oxide is probably the most common one, but propylene oxide, beta-propiolactone, chlorine dioxide are some others that are out there. Gases are little tiny molecules, so they're very good at penetrating through substances. Kind of a downside, you have long exposure times, 4 to 18 hours for sterility. And they work by cross-linking molecules and denaturing proteins, denaturing DNA, screwing stuff up. That's typically how most of these things work. All right, so I'm going to go ahead and cut that video clip off here. For the next video clip, we'll continue talking about some of these different chemical agents that are important for microbial control.